Today's session is structured in two major parts. First, we'll do a recap of the 9 sessions that we've done in a fun, engaging style. Okay. Microbe Express for Kids finally reaches its final station. And then in the next session, we'll do a mini quiz. So this is priming you up for the actual family quiz on June 3rd. Okay. And then finally, we're going to do a giant Mentimeter and enter all the words that we've sort of come across. All right. So are you ready for the recap? Are you all ready for the recap? Again, can I have some emojis in the chat window? Can we keep this a little active and live? Some fun emojis in the chat window. Very nice, Pavni. Thank you. We are getting there. Excellent, Chetna. Okay. Thank you, Kian. All right. So we are getting there. So let's do the recap first. So in our first session, Shreya, can you move the slide? So in our first session, we talked about microbes being everywhere. This was a session done with Snehal and me, the TTAS co-founders. We said microbes are, in water, in air, in soil. So where do you find microbes? Where all can you find microbes? I'll type the first one and you go ahead. So I'll say air. Where else do you find microbes? Water, excellent. And we talked about some waterborne pathogens or bacteria. All these slides are on the website. So you please look it up anytime. Body, yes, great point. Everywhere. Okay, cholera, soil, excellent. So all the words are coming back to you. In our surroundings, wonderful, wonderful, wonderful. So they're all coming back. And we gave you examples of microbes found in air, water, soil, our body, how they can be harmful to us, how they can be helpful to us, how they can be helpful to the environment, bioluminescent animals. Excellent. Food, some really great answers, guys. Some really great answers. Okay. So in the next slide, what we did there is we did a weekly activity. And I'm not sure how many of you did the activity and sent us in, but they are on our website. So you can go back to them anytime. And we said, what are the microbes found in these different places? So we said in soil, there are bacillus and pseudomonas. In food, which is on the picnic mat, there is salmonella and lactobacillus. On our body or skin, there's staphylococcus and corynebacterium. In water, there's E. coli, vibrio. In plants, there's agrobacterium and xanthomonas. In air, it's mycobacterium and aspergillus. So all these weekly activities have been prepared by Shreya. They're a lot of fun. So over the summer, when you have time, just quickly watch the session and do the activity. It'll give you something to do for 1 hour. Okay. So this was our weekly activity to tell you microbes are found everywhere. And these are the different microbes found in different places. All good? So are you going to go back and look at session 1 again, where we all started? I'm going to type a big yes. Yes? Okay, so maybe yes. Great. Thank you, Arshia. Okay, so now I will start asking more questions so that you get a chance to type in the chat window. All right, so in session 2, what did we do in session 2? In session 2, we talked about, Shreya, you can move the slide. Oh, sorry. Okay, before we get on to session 2, thank you, Kavish and Pooja. Let's look at our results from the Mentimeter poll for session 1. So do you notice a difference between the before and after? Do you notice any difference? I think they're almost similar. I think the kind of words and terms we saw. But what did we see with the emoji results? So Shreya's put together all these results so that we can see them together. We saw that before the session, people had responses like angry, worried, scared. After the session, the responses shifted to starstruck, mind blown, and less people were scared. Two people were scared. At least angry, worried moved to scared, we can say. Less people were angry or worried. But more people said they were starstruck or mind blown. Isn't that amazing? That after the session, people started getting more starstruck and mind blown by microbes? Yes? Okay, sounds good. All right, so now we move on to session 2. In session 2, we said yummy. We talked about microbes in food with Dr. Lorna Lancaster from the UK. So what did we learn about in this session? I remember learning a lot about cheese. What other words do you think you came across? There was a lot of cheese, there was cake, there was curd, there was also about like food preservatives. If you milk, bread, food preservatives, certain compounds that can help keep food free from bacteria. I think we learned a lot about that, and fungi also, bacteria and fungi. We also learned about this meat substitute called corn, which I had never heard of before actually. So you can go back to this session if you want to find out more about that and how microbes play a role in making all these foods. So we learned about how microbes help in making food flavorful, colorful. We also learned about how microbes help in preserving food. And I think that was very exciting about this session. Absolutely. Soya, Kian says, and Bhavik says pasteurization, preservatives, mold, curd making, lactobacillus. So we have lots of words. Okay. So let's do the activity together then. The next slide. So Mentimeter, we can look doing the activity together. So what's the bacterium found in these different cheese? And this is not an easy one, but at least let's do one of them. The blue cheese, which is this Roquefort, is actually a fungus, Penicillium roqueforti, that gives the blue color and texture and flavor. And all of the other cheese are also produced by the presence of some microbe, either a bacterium or a fungus. So take a deep breath and think about this. When you eat cheese, you are actually eating a microbe along with it. And these microbes are good for us. They've actually made the cheese what they are. Okay? So cheese to that. That's all we can say. All right. So this is another activity you can do at your own time. Let's look at the Mentimeter and the emoji results for this session. So Mentimeter clearly shows that suddenly words that came after the session were like food poisoning, curd, lots of things that you came across, ethanol. So new words that came across after the session. And the emoji results clearly show that people were less angry, scared, or worried, and more starstruck and mind blown, which is great, which is wonderful, right? So that was session 2 about food. Again, when you're hungry, sometime during the summer, this might be the one you go back to. Okay. Then session 3 was about social life of microbes, biofilms. We've talked about biofilms a lot on Talk to a Scientist, but we can't complete a Microbe Express for Kids session without actually having a session dedicated to biofilms. So this is why this was in. This is again with the co-founders, and we talked about how bacteria actually don't live in isolation, but have very active social lives. So do you have a social life, or is that a touchy topic? Do you have a social life? Yes, no. I'm going to type yes because I think I do. You guys think you do too? Well, that's good. You probably really actually do. And with grown-ups, is probably we think we do. Okay. All right. So we talked about how bacteria in populations do many things that they can't do when they're alone. Can you list 3 new terms about biofilms that you learned from this session? Any 3 new terms? Um, any 3 new terms? Arshia, Arhan, Kavish, can you type any 3 terms? Okay, one of you just type biofilm. Let's start with that. Yes, thank you, Bhavik. Biofilms. Then what else did we learn about when they work in communities? Bhavna also says biofilms. Yeah, I think we came across the word plaque. Excellent, Bhavik. Excellent. Very good, Kavish. So biofilms, plaque, we have 2. Any other? Anything else you can think of when it comes to bacteria and biofilms? I think one word we can say is matrix because building a community. Excellent, excellent. Thank you. We're done. Okay. So then moving on to the weekly activity we did for the social life is we asked you to talk about the microbes that are found in these different biofilms and are they helpful or are they harmful? So in oil spills, you have the bacterium Pseudomonas putida, which help in digesting hydrocarbons in oil. So if you have oil spills of coasts where there's a lot of oil mining and drilling, this bacteria in the water helps to break up hydrocarbons and therefore clean the oil spill. Exactly. Then you have kombucha tea. So in a very different context, here you're not in an oil drilling area, but you're sitting on your couch trying to be healthy, and you have kombucha tea. which contains Bacillus coagulans, another bacterium, which helps to ferment uh, the tea and break down certain sugars in the tea. Basically, it allows fermentation of the tea, which gives it its characteristic taste and also some other properties the tea has. Okay, so these are the kind of activities you can do once you've looked at the session. From oil spills to kombucha tea, right? Bacteria and biofilms are helping us everywhere. In fact, in kombucha tea, if you see, the biofilm forms right on top of the tea. You can actually see it. It's a thick layer on top. Okay, that's called biofilm mat or microbial mat. All right. Okay. Um, so the what Mentimeter before and after doesn't look super different because I think we've talked about biofilms a lot over the course of TTAS, but most certainly our emoji results seem to have moved. People are not just mind blown, but now people are actually happy, astonished, and starstruck. So you also see people are happy about microbes and biofilms because we've taught you a lot of the good things that microbes and biofilms do. Okay, correct. All right. That was session 3. Coming on to session 4, where we talked. This was a very fun session. In this, Nizam took us through the microbial lab in real time, with videos. So, can you remember some new equipment or lab tools or lab supplies you learnt of in this session? Some of them are right there for you to see. And while you're typing in, I'm going to charge my laptop. Just a second. Yes, changing of the tip. Absolutely. What else? What else? What other tools or equipment did Nizam show us in this session? Pipette, okay, gloves, incubators, serum. That's amazing, guys. You remember a lot. That is what else? Let me see. Let me see. Let me see. Okay, yes, serum, lots of microscope. Excellent. That's right. We also did see a microscope. So this is a very, very fun session. The slides and videos will be reaching you soon, and then you can watch this session. And so Shreya, these videos haven't been posted yet, right? But we'll be
No,
posting
not
them
yet.
over the course.
Yeah,
Great,
will
over the
be.
course. Yes. So it's a very electron microscope we talked about. Excellent. Okay, so the next slide will tell you what activity was based on this. And this is going to be fun. So can you look at this activity? And again, a very nice activity developed by Shreya. And say what is wrong with how this person is working in the biosafety cabinet? What are the mistakes that the scientist is making that is not with good lab practice? Can anyone point them out quickly? Everyone likes to point out mistakes. So let's do this like full flow. What are the mistakes? No mask. Very good, Arshia. No mask. What else? 6, 7. Okay, yeah. I noticed that she has makeup inside the cabinet, or this person. I noticed that there is some chocolate wrapper. What else do you notice is wrong? A mobile phone, which you shouldn't. What else? Not clean. Her, I don't know what's the problem with her eyes. I'm not sure about that, but cluttered. But so there are lots of mistakes. So after you see Nizam's session, you can go back and do this, and you'll be able to point out all the mistakes or all the practices that were not according to good lab practice. Okay. Then what happened with the session results? Let's look at that. Avisha says all of them. We agree. Okay, we agree. So when we say the session results, um, so lots of new science terms came across in the after. You can see the crystal violet, fridge, agar, pipette, petri dish, lots of more technical terms, which was the whole idea of that session. And people again really shifted to being very starstruck and mind blown by microbes about working in a microbiology lab, which is also great because that's what we want to, expose you to. We want you to think about or see parts of microbiology and science that you don't just otherwise see. So we actually took a dive into the lab. It was a lot of fun. You can go back to this session. Okay, so session five, at this point, we were halfway through the journey and we decided to do something extreme. So we talked to Dr. Aparna Banerjee about extreme environments and microbes that are found there. And this was a really fun session because it took us to places on Earth virtually, vicariously, that we haven't probably visited. So, what are some extreme environments on Earth where you find microbes? There's one really easy one which I will not type because I'll give you a chance to type it. Excellent. Antarctica, Chile, volcanoes, hot springs. Okay, so I will type one. Hot springs and space, where we don't know whether we have microbes, but very likely we do. Mars, Yellowstone Springs, excellent. So in the next slide, you will see the activity based on that, which was very similar to the question that what are these extreme places? So this is Pink Lake, Australia. This is Yellowstone. Three is Antarctica. And four is a volcano lake in Costa Rica. Costa Rica is Central America. And so these are places on Earth that are known as space signatures. That's what Aparna told us, that they are so extreme that in their conditions, whether it is temperature, pressure, under in volcanoes or heat, Arctic, that microbes found there are the resemble if microbes were found in space. They resemble that situation, and therefore studying microbes in these environments on Earth help us to understand how and where microbes could survive in space. So these were space signature regions, and Aparna has worked in two of them, one in India and one in Antarctica, Chile. Okay, Antarctica off the coast of Chile. Okay, great. So what was the results of this session like? What were the results like? Um, Not knowing what to expect, the results of the session, again, we got, I think, a good mix of technical terms like biosignature, astrobiology, and other terms. But importantly, most people, yes, again, people were, you saw the shift to the left where they were very starstruck and mind blown and um, happy. Okay, about they were amazed rather to put it all together. Okay, then we move on to session six. So after extreme environments, we came back to session six, where we talked about microbes and infections with Holly Shaw, again from the UK. So you can see in this season, we've really tried to get speakers, a good mix of speakers from India and out of India. And Holly Shaw talked to us about microbes and infections. So from extreme environments, we came to microbes in and on our body. And she went through different infectious diseases: bacterial, viral, fungal, parasitic. So, can you name an infectious disease? Any one? I'll name one, which is easy. Chickenpox, excellent, Kavish. I'll name malaria. Cholera, says Bhavik. Plague, excellent, Arshia. Ebola, corona, excellent. Look at your level of knowledge, wonderful. So, in the next slide, what was the weekly activity based on Pitrasis alba, E. coli, smallpox, excellent. We gave you pictures of different microbes and asked you to identify them. So the first was a virus, and we asked you to give some examples. Second is a parasite. Third is a fungus, and fourth is a bacterium. And you can watch the session, see the slides, and go back and do this activity. And you will have all these answers on your own. All right, so what were the results of this session like? So when we took our session polls, we saw again a wonderful shift that you can see. You can clearly see people are less worried, angry, and scared, and more astonished, more mind blown, or at least more astonished. And they were also less confused, which is a great sign. So that means Holly's session worked. And again, before and after is not too very different because we've also talked about microbes and infections a lot. But you clearly see the shift with the emojis, which is wonderful. Okay, so now coming to session 7, 8, and 9, before we enter the mini quiz. The mini quiz is what you guys are going to be doing with us. So, session 7, we talked about the gut microbiome. And if you remember this session, Dr. Sunil Nagpal had done a wonderful job with the creatives for this session, right? And he took us through where microbes are found in our gut and also why we need microbes in our gut. So, can you tell us some answers? Why do we need good microbes in our gut? What do good microbes in our gut do for us? Let's see if we remember the session without going back. What do good microbes in the gut do for us? Excellent, Pavni. Digestion, They help build immunity. What else? They also produce some stuff. They break down food, excellent. Digestion, the process of digestion. They also help make a certain vitamin. Can you think of what vitamin that is? B12. Okay, yes, yes. Vitamin K, excellent. They help make us make certain vitamins. So actually, microbes in our gut are just not, there. They're not passive entities. They actually help us. They help us in many ways. They also here help protect us from bad microbes that can cause gut infections. So lots more about that in a very creative style in the slides, which you can go back to. So before the session, most people will think of gut microbiome or microbes in poop as yuck. We gave you an activity to do before, after the session, and this was a very nice fill in the blocks, and the answers are what we've given you here. You can also do this activity and check the answers because these slides will also be up on the website. Um, but post activity, we did the analysis, and from yuck, which was microbes in poop, we went actually very shockingly. Most people were starstruck and mind blown. And it sort of stayed that way. And the worry, angry, and scared didn't show up much, which means that even after the session, people sort of continued to be starstruck and astonished, mind blown by the microbes in our poop. And we have a new respect for them now that we know what they do for us. Okay, that was a great session too. And now coming to session 8 and 9. So in session 8, we saw microbes, different microbes under different microscopy techniques with Sophie Dach. And can you describe how microbes look under the microscope? Can you describe their shapes? Can anyone of you describe their shapes? There are some clues in this figure itself. How do microbes look under a microscope? Irregular, excellent, excellent. Diverse, excellent. Filamentous, wonderful. And that's exactly what the weekly activity was also about. Spiral, wonderful. Sphere. So moving on to the weekly activity, and you guys have all the answers. Flagella like, okay. We gave you these pictures and asked you to fill it. So the first one are rod shaped and they're known as bacilli. The second are spherical and they're known as cocci. Flexible spiral are spirochetes. Rigid spiral, like a corkscrew, is spirilla. Corkscrew or someone with curly hair, you pull it and it like bounces back. That's spirilla. So you can watch the session, see the different microbe shapes, and come back and do this fun activity. Okay. So as you see, we've always tried, or rather Shriya has always tried to make the activity relevant to the session so that you're like sort of on a journey. You're truly like on a journey and hitting every station and doing a little activity as you go. Okay, so what happened with the results? What happened? Dog tail like, okay, oval, no membrane, very good. Again, people were very positive, astonished or mind blown. But we could again see a huge amount of technical terms show up in the Mentimeter poll, which is great. Okay, finally, session 9 was about antimicrobial agents with Blessing Anoniye, who was also from the UK. It's very interesting. We had all our guest speakers have some connection to the UK. And this was by some part of design because the Microbiology Society that funded this season of TTS is based in the UK. So Blessing talked to us about different antibiotics, antimicrobial agents. She also talked about antibiotic resistance and the emergence of something called resistant organisms or superbugs. So do you remember what are superbugs? Can anyone recollect what are superbugs? Are they microorganisms that live on Superman? At least say yes or no. Bugs that get to be stronger. Okay, they do get to be stronger. So that part is correct. Yeah, no, thank you. Thank you. Thank you so much. So they're basically bugs that are resistant to lots of different antibiotics. So if you get an infection with them, they're difficult to treat. And one of the reasons we want to be careful about our use with antibiotics, exactly, resistant to antibiotics. Very good, Bhavik, is so that we don't get superbugs. So we always have antibiotics against the bugs that we get infections with. Okay, so this session has a lot more on that, but let's look at the weekly activity. And we asked you to classify or name the classes of antimicrobial agents. And we talked of those that inhibit viral uncoating, they are antiviral. Those that act on the cell membrane with a certain type of cholesterol called ergosterol are antifungal. That inhibit the synthesis of the bacterial peptidoglycan cell wall are antibiotic. And then finally, antiparasitic, that paralyze parasites like roundworm, tapeworm, etc. So you can go back to this as well. So let's go to the weekly activity. Let's go to the activity, the poll results. And Chetna correctly says strains of bacteria resistant to several types of antibiotic. Again, we saw lots more technical terms apart from ha 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 ha, ha, which probably in the context of antibiotics could be technical. Nothing, it's not a laughing matter. So we agree with the term. But while we had a less of a poll result, we overall say that they are, people were mind blown, people were starstruck. Okay. Now, so this was a quick recap. I hope you've enjoyed it. Lots of information for you to work on over the summer. Now, all previous sessions are going to be there on the website. We're going to launch into our mini quiz. So, are you all ready? So, we're going to start this mini quiz once we get lots of thumbs ups, okay, in the chat window. So, you have to be ready to start this mini quiz, okay? And we need to start seeing the thumbs ups to tell us that we are ready. Okay, so it's been a question and answer. Most of them are multiple choice. Okay, thank you, Chetna. And a few, one or two of them are not multiple choice. But we'll go through them one by one. Okay, so are we all ready for question one? Yes, all right, let's get started. Question one, Shreya. Question one. Everyone ready? Okay. You're going to type the answer in the chat window. Identify the shape of the bacteria in the microscopy image. That means the one on the right. So first you see a colony, then you see bacteria in liquid in a tube, and then you see bacteria stained and seen under the microscope. So are they bacilli? Are they cocci? Are they spiral forms? Or all of the above? Okay, cocci. We'll give everyone else a little bit of a chance. Cocci, D, all of the above. Okay, take a closer look, guys. Take a closer look. Okay, the answer is, going on to the next slide, the answer is cocci. What you see are actually circular forms of bacteria. All right. And if you see them a little long, it's because of two, three of them have just lined up. So the answer is cocci. Okay. Well done, everyone. Okay. Now, everyone ready for question two? Yeah? Some thumbs up? Okay, great, great, great. Everyone's ready for question two. Moving on. This image, this is a tough one, guys. This is a tough one. This image shows colonies of bacteria growing on a plate which contains protein. It also shows a group benefit to the bacteria growing together. So this was, think of our biofilm session. Some benefit to the bacteria growing together. What is this benefit? In addition to seeing bacteria on a plate containing protein, you see something else. This is a group benefit because many of these colonies growing together allow this benefit. What could be the benefit? Well, very good, Abhishek. Protection against antibiotic is one group benefit, but that's not the one we see on the plate. This is a very good answer. Excellent answer. What is the group benefit? Okay, so they're able to acquire food. Great, you're on the right track. Not quite resistance, Kian, but that's also a group benefit. What do you see around the colonies of bacteria? Do you see something unique? So, not horizontal gene transfer. What you see is halos, so where bacteria have produced enzymes that have digested the protein, and the digested protein are now being used, can now be used as food by all the bacteria on that plate. So that's how it's a group benefit. It's a benefit to everybody. Anyway, you can visit. This is a tough question. I told you right in the beginning, but you can revisit this. So this answer is public goods. It's basically every bacteria produces some amount of enzyme that digests the protein and allows all other bacteria to use that digested protein. Excellent, Bhavik. Excellent. Okay. So moving on to question three. This is an antibiotic susceptibility test. Okay, this is a relatively easy one. Is the bacterium on the plate treatable with antibiotic A or B? Which antibiotic will you give to treat this bacterium growing on the plate if it was in your body? Very good, Bhavi. B, B, Avishai, B, Pooja, B. Lots of B's, lots of B's. And why B? You're all right. You are all right. But just to make you think a little more, why B? Why B? What's different about B? Yes, Bhavik, excellent. You see a zone of inhibition. Well done. That means that antibiotic in B was killing the bacteria around it, and therefore the bacteria is susceptible to the antibiotic, and you'd give that antibiotic to treat. So, excellent. This is how we test for antibiotic susceptibility in the lab, and you have correctly identified the results. Excellent, Kian. Circle around. Very good. Okay, so now question 4. Okay, this is an image of some microbe. This is a chemical reaction that the microbe can um, mediate. What do you think? What delicious food does this microbe and chemical reaction produce? Is it cheese? Is it bread? Is it meat substitute? Or is it corn syrup? What is the microbe? And this is a chemical reaction. And together, what delicious food do they help us produce? D, okay. Anyone else? Not sure about corn syrup. What is this microbe? Can anyone identify this microbe? Yes, yes, Bhavik, it's yeast. Well done. And the answer is bread. Well done. So this is yeast. This is a budding yeast. You can see it's forming little buds from the yeast. It's called Saccharomyces cerevisiae, and this helps us make bread. This is the reason bread rises. So well done. Because of the carbon dioxide produced, bread gets holes and it rises. So this is a fungus, Saccharomyces cerevisiae, helps us yeast, which helps us make bread. We're very good, everyone. Very good. Okay, so coming to question 5, we are halfway through the quiz. Can you believe that? And we're right on time. This glue holds bacteria together in a biofilm, and it consists of what? Protein, carbohydrate, water, all of the above. Protein, okay. Bhavik says all of the above. Okay, true, true. So this biofilm matrix or glue contains everything: protein, carbohydrates or sugars, and water. So all of the above is the answer. And that holds the biofilm together. Whether it is in a lab where you see biofilm growing like these little mushrooms, or it's in a human infection where you see bacteria growing in clumps, and that's what you call the biofilm. It looks like moss, but it's actually a bacterial mat. It looks like moss. So yes, this matrix contains proteins, sugars, and water. Okay, well done, everyone. Well done. Coming to question 6 now. This one you all should answer, and it's Mother's Day this weekend. And so you have to go and say thank you to your moms for this. All right. Name this bacterium. It's a baby's first friend. It's a gift from your mom, and it's found in our gut. All right. Everyone says C already. Yes, 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 yes. Very well. This is bifidobacterium. It's called bifido. Bifido means two, right? So it's because it's shaped like a little Y, and it's the first microbe that's found in your gut. It's found in your first poop. In case you thought you were like super clean when you were born, you were not. Do ask your mothers about that. And it's actually a microbe that your mother gives you. So this Mother's Day, it would be very appropriate to make a card which says, you draw bifidobacterium and say thank you for bifido, and your mom will be like, what is this? And then you can tell her all about what you learned at TTAS, okay? And say, you gave me so much, mom, including a bacterium. That could be a tagline for the card. All right, sounds good? Yes, so this is bifidobacterium. It's the first microbe found in babies, found in your gut, found in your poop, and it's a gift from your mother at birth. Okay, all right. Moving on um, to question 7. All the following are space signature regions with microbes in extreme conditions, except, we talked about this: Amazon, Antarctica, Campanario, Chile, or Panifala, India. All of these are space signature regions, except, brilliant, brilliant, brilliant. Look at it. Look at that. Except the Amazon, because Amazon is a rainforest. The last we checked, we don't see rainforest regions in space. So moving on to the next slide. Yes, the Amazon is not space-like, but Panifala, India, which is where Aparna really originally worked, Campanario, Chile, and Deception Island, Antarctica are all signature space regions. So not all scientists work in labs. Some scientists do their science in such extreme environments. It's whatever works for you. All right. So you can also bring your choices and individual personality into the science you do. If you're someone who likes adventure, likes being outdoors, then this is the kind of science you should do. All right. So question 8. We're slowly coming to the end of the quiz. Everyone's done really well. But let's do question 8. The structure of the bacterial cell that the antibiotic oh my god. I haven't even finished. It is the structure of the bacterial cell that the antibiotic penicillin acts on is the cell wall. Excellent, excellent. Oh my god, too many A's. But it is the cell wall. Penicillin. So here you see a bacterial cell and different antibiotic classes, and you see penicillin right here in blue that inhibits the formation of the cell wall, and then the bacteria cannot grow and it essentially dies. So well done, guys. Well done, all of you. Okay, coming to question 9, the second last question. Name this laboratory equipment or tool. Is it a pipette? Is it a petri dish? Is it an incubator or is it a shaker? I mean the whole thing, huh? Is it a pipette, petri dish, incubator, or shaker? Lots of C's, lots of C's. Correct. It is moving to the next slide, it is an incubator, which helps maintain a temperature of 37 degrees because the human body is at 37 and we want to study microbes under conditions that mimic or resemble the human body. Correct, correct, Bhavik. Well done, everyone. Well done. You guys are ready for a career in microbiology. And finally, the last question of the mini quiz. I hope this preps you for the family quiz. All of the following are virus caused infections, except. Now you read the options. Lots of different answers. All are virus caused except tuberculosis. Correct. Tuberculosis is caused by a bacterium, mycobacterium. The name says it all. Mycobacterium tuberculosis. Well done, everyone. Well done. I think it's been a great result, Shreya. So many questions were right. So again, you can go back to all the slides and do this quiz again if you want. But this is how our family quiz will be, except the questions will be tougher, and you will be doing them with a family member. Okay, but they will be a little tougher questions. All right. So are we going to see you at the family quiz on June 3rd? Hands up, the thumbs up if you're going to see you at the family quiz on June 3rd. Awesome. Who else? Yes, yes, yes. Oh, wow. So start talking to your family members now. Start going through TTS slides, videos, start talking science, thinking science, and you'll be good to go. And we've always had very fun prizes for our family quiz. So do show up. Okay, we look forward. Thank you all. Thank you all. We look forward. Okay, so now we finished our first part, which was the recap. We finished the second part, which was the mini quiz, which was okay. Avisha says maybe, but do try and come, Avisha. We love the honesty. Okay, so now if you could all please go and do the giant Mentimeter cloud together. So let's go to Mentimeter. I will pull it up here and you guys start typing questions. Now remember, we want a giant cloud. So we've allowed you to type 10 answers each. So take your time and let's do 10 words each. Okay. And the question is, what words or microbe related words did you hear of or see or learn of during Talk to a Scientist? So, Shreya, I don't see anyone typing it in. Are you guys typing it in?
You could give us a thumbs up once you're done.
Pardon?
I also don't see. Maybe you have the access to it.
I do, but I don't see anyone typing in. Have you started typing in, guys? Oh, yes, Kavish? Okay, then why am I not seeing it? Okay, the link is working. Yes, Bhavna is also, Abhishek is also typed. Okay, yeah, now I see words coming. Okay, okay. Go ahead. 10 words.
Maybe you could share the screen.
Yeah. I'll share just a minute. But I'll see. This is all I see. Can you see the screen?
Yes, I see. Yes.
Okay. So, all right, let's take our time. So, first word that showed up is resistance. Okay. Any other words?
Maybe try refreshing it.
Yeah, even I'm thinking. Okay, microbiologist. Okay, that's another big word that showed up. Great. Oh, yeah, see, suddenly. Lots more words. Wonderful. So let's work. Everyone, let's try and get 10 words in, okay? Microscopic, unicellular, lab, bifid, chile. Okay, very nice. Very nice.
I see bioluminescence.
Bioluminescence, exactly. Yeast, Deception Island. Okay, wonderful. So let's see, everyone has to, let's try and get 10 words each, everyone. So can you tell us how many words you've typed if you finish typing? Let's really try and get 10 words each. Oh, lots of words. Antiparasitic, antibiotic, biosignature. Safety, I love that word. Safety, love it. Cockeye, micropipette, hungry. Yeah, that's true. Oh, Kavish has said he's done 10. Thank you, Kavish. Thank you. Okay. Anyone else? Avishai says 10. Excellent. Can we have some more words? Rod shaped, Mycobacterium tuberculosis. Excellent. Lab. Okay. Very nice. Kavish wrote hungry. That makes sense. Wonderful, Kavish. Wonderful. Okay. Any more words, guys? Maybe we type a few more. Let me try and refresh this. Excellent. It's shown up again. Can we type a few more? Arshia says 20. Oh, great. Okay, maybe everyone type one more. Let's try that. I don't see any new words coming in. Anyone wants to type any new words or shall we stop? Whatever works. Thank you. Lots of words. Bacterium poop, microscopic petri dish, incubator. Very nice words. Okay. All right. Okay. Then we can, shall we stop it or anyone wants to type? Okay. Then I think we will stop it. Thank you very much. And we'll go back to the slides, Shriya. Thank you all. That was very nice of you to type so many words. Thank you. Very nice. Okay. Now let's go. All right. All right. And now the moment of reckoning. So Shriya, we are done, right? I think we're
Yeah.
done. Okay. Yeah.
Uh,
Bye everyone. See you June 3rd. No, we actually have our raffle prize coming up. So this is going to be a fun raffle prize, and she has already done it so nicely. So many of you, let me tell you how this worked. Many of you entered the weekly activity and sent us an email. Yes or no? Yes. We said everyone, the person who is eligible for the raffle entry has to have done all the activities. But we realized that was possibly not possible. You had school and other things. So we said, even if you've submitted one entry, you get to be on this raffle. So these are the names. We have Deepak Singh. I don't know if you're here, Deepak. I don't think so. Bhavik. Bhavik is here, right? Bhavik, you want to raise your hand? We have Ananya Tonapi. We have,
one
okay,
second. Yeah, I could. Yeah, here's the list of names.
yes, just a minute. Just a minute. Okay, 1 minute, Ashriya. 1 minute. Just a second. Okay. So, Shreya, can you just say the names and we'll see if they're
Uh,
there. So, Bhavik is there, which is great.
I see. We have Ananya Sonapi,
Anand.
Bhavik,
Okay. Hmm.
then Deepak Singh,
Okay.
Gracie Duggar,
Okay.
Mohak Chandra, Narendra
Okay.
Yadav, Pooja S, Rishabh Ankur, Sahan Rajkumar, Sayan Rajkumar, Veda Kalpi, and
Okay.
Vijeshwari I'm sorry, I am not able to. So
Okay.
we
No
have
worries.
around like 12 entries.
Fantastic. So we do have 12 entries. Is there anyone who sent a weekly activity, but this name is not there? This is the time to make us change it. Did you send in a weekly activity? Anyone whose name is not there? Okay, so no, right? Final call, guys. Did you send a weekly activity and your name is not? Okay, fine. Okay. So now, Shreya, so the prize for the raffle is a physical copy of the hands-on book. So after we spin the raffle, we will get in touch with you by email and post you a physical copy of the book. But of course, everyone has access to the ebook. It's on our website. Okay. So I hope you're enjoying that. That's another thing you can do over the summer. Okay. So
Ready?
yeah, how are some people entered twice?
Uh, no, I don't see twice.
Oh,
It's just
Sayan
one.
and Sahan. Okay, okay.
Yeah,
Different names. Okay,
yeah.
different names. Okay. So okay,
Ready.
1 minute. So we are all very ready. So no, this is the final call. The prize is a physical copy of the hands-on book. Shall we spin? We try to do this in this fun Wheel of Fortune way, and Shreya has really set it up so beautifully. Thank you, Shreya. Let's take a screenshot of this. Um, no. Avishai says, what if they are outside India? We will post outside India. Don't worry. We ca- we haven't figured out posting to space, but anywhere on Earth we will post. Okay. All right. All right. So are we all ready to spin? Are we ready to spin the wheel? So everyone, thumbs up, thumbs up, thumbs up, thumbs up. Come on. Let's get this going. We want some thumbs ups in the chat window. Thank you, Avishai. Thank you, Chetna. Mohak. Isn't this exciting? Finally, Microbe Express is coming to an end. The raffle prize is being given out. And we're planning our summer at TTAS. It's been such a great session with you guys. Really great. You've been very engaging, very interactive. Thank you so much, all of you, for joining. Okay, Shreya. So now let's move to the spin the wheel and find the winner of the physical copy of the hands-on book.
Okay. One,
Hmm.
2, 3.
Well, congratulations to Rishabh. Shreya, just a second. Let's just take a picture of this.
Yeah.
Done. Congratulations to Rishabh. Congratulations to all of you. While Rishabh gets a single copy of the book, there are many more prizes to be won in TTS over the summer, including the family quiz. So we want you all to be there.